I'm getting a lot of questions about requests for regrades of exams. So let me just say a couple things about that. Biggest concerns are, what does it mean for fishing for points? Okay? So I have a fair number of people in the course who think that when I give an exam back, that automatically means you return it in for regrade. Okay? Because it's a way to get points. And that's not what this is set up for. This is set up, if there's a mistake that's made in the grading of your exam, then you turn it in, you make a note of what it is, and we deal with that. Okay? And if you're just turning in exams to get regraded, or you're not making reasonable cases for what's there, that's fishing for points. Okay? So you want to be careful with this. This is a mechanism for dealing with errors. Okay? That's what it's there for. Okay? And yes, I do see people fishing for points, and yes, I do take points off when I see that. This is not a mechanism to just get exams regraded and see if you get more points. Okay? So please understand that. Okay. That's it. We move on to our discussion of carbohydrates. So we finished up talking about, last time, blood clotting. And today, we turn our attention to carbohydrates. So most of what we'll be talking about here are structural considerations. But because there are structural considerations, that leads to other things about them that we'll find will be important. A lot of what I'll talk about today are things that you've seen in organic chemistry. Okay? This is probably one of the easiest to understand lectures of the term, which is a cause for celebration. And a lot of it is going to be review for you. Okay? The importance of structure can't be underestimated. You've been hearing about structure and function the whole term. And this carries all the way down to the level of individual carbohydrates as well. So when we talk about carbohydrates, one of the first things we have to keep in mind is nomenclature. Okay? So there's a pretty big body of nomenclature that's used to describe carbohydrates. And as I say, if you're going to walk the walk, you've got to know how to talk the talk. So being able to understand the nomenclature and discuss the nomenclature intelligently is important. All right. Well, the first nomenclature I'm going to give you, carbohydrates, of course, the term carbohydrate actually tells you what the structure of the molecule is. Carbo referring to carbon, and hydrate referring to water. So if we look at the chemical formulas for carbohydrates, for example, glucose is a carbohydrate, and glucose has the formula C6H12O6, which can be written as CxH2OX, right? Carbohydrate. That's where the term actually comes from. Another term we use to describe carbohydrates is a saccharide. A saccharide is something that is another term. It's equivalent to carbohydrate. And saccharides come from, I believe, Latin, and it means sweet taste. Right? So when you hear something described as a saccharide of some sort, that's what it's referring to. The first nomenclature I'll give you is to remind you that for the monosaccharides, and when I say the term monosaccharide, that means one sugar. All right? For monosaccharides, they come in two general forms that we can talk about. One form is known as aldoses, and the other form is known as ketoses. Okay? So if we look at these on the screen, we see, excuse me, we see, first of all, a ketose. And we can recognize a ketose by virtue of the fact that it has a ketone. And it is a carbohydrate because its structure is C3H6O2. Or I'm sorry, O3. C3H6O3. All right? So that's the equivalent of the carbohydrate structure that we've seen before. Okay? And the other form, so this is the ketose over here. The other form that we have of a carbohydrate is that of an aldehyde. Okay? So this guy's a ketose. Over here we see aldehydes because there's that oxygen on the end instead of being in the middle. These two guys on the right are what we refer to as aldoses. Okay? The structure on the left has a name. It's called acetaldehyde. Okay? It's not acetaldehyde. What am I saying? It's not acetone. It's dihydroxyacetone. Thank you. I didn't get enough sleep last night, I think. Dihydroxyacetone is one of the simplest carbohydrates that exists. The ones on the right are both known as glyceraldehydes. Okay? Now, you'll see there's two of them. And the reason that there's two of them and there's only one of this is that the middle carbon of the glyceraldehydes is asymmetric, meaning it has 4 different things attached to it. Now, it's here where you guys are going to like biochemistry better than organic chemistry, I hope, okay? Because we keep the designation of these structures very simple, okay? Very, very simple. We will refer to structures as D and L, and I'll show you the simple rules for those, and that's basically what we're going to do, okay? Now, this guy only has one form because that middle carbon only has 3 things attached to it, not 4. All right. So, aldose and ketose refers to general categories about whether they are aldehydes or whether they are ketones. All right. We can also categorize the saccharides by the number of carbons that they contain. Okay. So, something that has 3 carbons could be described as a triose. Something that has 4 carbons can be described as a tetros. Something that has 5, a pentose. 6 has a hexose. 7, which you hardly ever see, as a heptose, although there are heptoses that are out there. And 8 has an octose, is an octose. All right, so I could describe something as an aldose, and I could also describe it as a triose. So that's what you saw, for example, with glyceraldehyde earlier. It was an aldose, and it was a triose. Okay? Glucose, by contrast, is an aldose, and it has 6 carbons, so it's a hexose. We could also call this an aldohexose. All right? An aldohexose. Aldo referring to aldehyde, and hexose referring to the fact that it has 6 carbons. Okay. So those are monosaccharides. We only see 1 sugar in them. We will see later disaccharides, meaning that they contain 2 individual sugar units. We'll see polysaccharides that contain many, many subunits. Okay. So those are some general terms. Here is a very nice diagram that shows a variety of other terms that we use to describe carbohydrates. So I'm going to go through these with you sequentially. All right? The easiest one to start with is right down here on the lower left. Okay? On the lower left, we have the term enantiomer. Enantiomers are molecules that are mirror images of each other. They're mirror images of each other. Okay? So we see D-glyceraldehyde and we see L-glyceraldehyde. The difference being the configuration of that hydroxyl group on the middle carbon. You'll notice that the D-glyceraldehyde has the hydroxyl group on the right, and the L-glyceraldehyde has the hydroxyl group on the left. Okay? So enantiomers refer to molecules that are mirror images of each other. Very, very simple to understand. Okay? Now, when we go to higher order carbohydrate structures, like let's say glucose and maltose, we discover that they have more than one asymmetric center. That is, if we count them, there's 1, 2, 3, 4 carbons there that actually have 4 different things attached to them. So there are 4 asymmetric centers within maltose. There are also 4 asymmetric centers within glucose. Okay? Well, if we look at these two, we could look at them very quickly and say, well, these guys are not mirror images of each other, because if this one on the right was a mirror image of the one on the left, it would mean that we would have to have a hydroxyl, 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 hydroxyl on the left of the other one, right? To be mirror images, they would have to match up like you put a mirror in there. Well, glucose is not a mirror image of maltose because we can see that it only has 1 hydroxyl on the left. All of its other hydroxyls are on the right. So we have a term for molecules that have the same formula, okay, and the same category, meaning that they're both aldoses, but that have different configurations around their asymmetric centers. They're not mirror images. This term we give to them is called diastereomers. Okay? So diastereomers, these two guys are diastereomers because they're not mirror images. They're both aldoses, but they differ in the configurations of those hydroxyls around their asymmetric carbons. Okay, so that's the difference between an enantiomer and a diastereomer. Okay? Yeah?
On the diastereomers, is there a typo because there's
7 oxygens. There is a typo, yeah. Good eyes, yeah, very good. This is brand new sugar we just made up here. You know, that's interesting. Book typos are always fun to find. I hadn't noticed that one before. I was author on a textbook back in about 10 years ago, and I got in on the third edition of the textbook. I mean, I told you this guy's a story, but I got in on the third edition of the textbook, and as I was going through it, I was seeing a figure that had appeared in the first two, and it had a carbon with 5 bonds to it. And I pointed this out to the two previous authors, and they went, "Oh my God, it's been out there for all this time." So, yeah. But yeah, you're right. That has one too many oxygens. Okay. So diastereomers versus enantiomers. All right. Now, I've already described these differences. I don't give this a name, constitutional isomers. I think that just gives you another term to remember that doesn't really tell you anything about them. So I don't use that term, but you obviously should know the difference between an aldose and a ketose. Okay. Another term that we use to describe these guys is epimers. All right. So epimers are two sugars that have the same general formula. That is, they're both aldoses in this case. They could both be ketoses as well. But gesundheit. They are both aldoses in this case, but they differ in the configuration of only one carbon. All right? So let's see. So one carbon would be right here. They differ in the configuration of this guy, carbon number 2, but they have every other carbon has the same configuration. So two sugars that differ in the configuration of only one carbon are called epimers. Epimers are a subset of the diastereomers, because these epimers are also diastereomers. They're not mirror images. Okay? Okay. The last term that I'll give you here that's shown on the screen is that which we call anomers. Okay? And anomers arise as a result of cyclization of the sugar. So sugars, as I hope you recall from your organic chemistry, can exist in a straight chain form, and they can exist in a ring form. And when they move into a ring form, something interesting happens. Okay, here's carbon number 1 in the straight chain form. You can see that it's an aldehyde. However, that carbon number 1, when it makes a ring, will link with typically carbon number 5. And when it links with carbon number 5, it makes something that looks like what we see down here. These guys down here have 6 members in them, in their rings. One of the members is an oxygen, which means that if we count the carbons, and I always recommend that when you look at these, you number the carbons as the very first thing you do. You number the carbons, you say, here's carbon number 1, 2, 3, 4, 5, 6. We see carbon number 6 is sticking up here apart from the ring. Okay? And that's because carbon number 1 interacted with carbon number 5 to make this ring. Oxygen is one of the 5 members of that ring. Now, why do I tell you that? Well, the significance of that is that by having carbon number 1 interact with this oxygen on carbon number 5, we actually create a new carbon that's asymmetric. We create a new carbon that's asymmetric, and that's carbon number 1. You see carbon number 1 up here is not asymmetric, but this carbon down here is attached to 4 different things. An oxygen, a hydroxyl, a hydrogen that's not shown, and another carbon. Okay? So we've created a new carbon that's asymmetric, and that new carbon is called anomeric. It's the anomeric carbon. The anomeric carbon will always be the carbon that was either the aldehyde or the ketone, as we will see. So the anomeric carbon will always be the one that was either the aldehyde or the ketone in the original structure. Okay, so that these two guys are anomers, meaning that they only differ in the configuration of their anomeric carbon. The anomeric carbon can either be in the up position, as shown here, or in the down position, as shown on the left. If it's in the down position, we call it alpha. And if it's in the up position, we call it beta. All right, so there's a lot of nomenclature there, a lot of things to get your head around in terms of thinking about structures. There's one more thing about nomenclature on this slide that I want to show you and describe to you, and then I'll move on. Okay? Any questions before I talk about this one last thing? Yeah? So when this guy goes from being a straight chain to be closed, it can go either way. It can be up or it can be down. So there's not a way to say it's going to be one or the other. Okay? All right. Now, the last thing I want to say has to do with the nomenclature D and L. So we saw over here, actually not over here, but when we talked about these guys up here, I told you that these can exist in two different configurations. One was in the D form and the other was in the L form. We need to talk about what that means. All right? So if we look at these guys here in the straight chain form, it's very easy for us to tell if they're in the D form or the L form. Here's what I was thinking of here. All right? Here's the L, here's the D. Well, it had the hydroxyl on the left, it had the hydroxyl on the right. Hydroxyl on the right referred to a D, hydroxyl on the left referred to an L. Oh my God, what do I say about this guy over here? Well, you'll notice that it says that they're in the D configuration. How would you tell it's in the D configuration? Turns out it's very simple. If you looked at the next to last carbon, that is, as you count down the next to last carbon, the last asymmetric carbon, if the hydroxyl is on the right, it's in the D configuration. If the hydroxyl is on the left, it'll be in the L configuration. Now, I need to clarify something with that before you take that too much to heart. Okay? If you have a sugar that has the hydroxyl on the right, the next to last hydroxyl on the right, it's in the D. If you have the next to last hydroxyl on the left, it's an L. So you might look at this and say, okay, well, I can take, here's D-glucose. Is L-glucose simply the same thing with the hydroxyl on the left? And the answer is, it's not. Oh, man, Ahern, you said it was going to be easy. All right? Well, it is easy. All right? L-glucose is the mirror image of D-glucose. Not only is this hydroxyl on the left, but this hydroxyl's on the left, this hydroxyl's on the right, and this hydroxyl's on the left. That's what L-glucose would look like. Okay? Now, am I going to make you go and draw D and L and rings and all that sort of stuff? Okay? I'm not. Virtually every sugar we're going to deal with is going to be D, but you should know, especially in straight chain, what an L sugar looks like. Yes, question?
Yeah, so you said that an alcohol is mirror image of the alcohol. Is that true for all?
It's true for all sugars. It's not just for glucose, but for all sugars. If I give you the D form of some sugar and I say, what's the L form, you should immediately think enantiomer. It's the mirror image of that. Okay? Is there another question? Okay. Yeah.
Will we ever deal with any ring-shaped L's?
Will you ever deal with any ring-shaped L's? You'll actually see one here. I'm not going to ask you to draw them and so forth, but I'll tell you what an L-shaped ring structure would look like. This carbon, instead of being up, would be down. Yeah. Okay? But for the most part, most of the sugars that we find biologically are in the D configuration. Most of the sugars that we find are in the D. Not all, but most are in the D configuration. Okay. So there's a lot of nomenclature there. Make sure you understand the nomenclature. So when I ask you on an exam about something, you can go through and do that. What you see on the screen here is a listing of common monosaccharides. Common. These are the ones that are probably the most commonly found in all of biology. All right? And your next question is, how many of these are we going to memorize, Ahern? All right? And the answer is, well, I don't ask you to memorize a lot of structures in this class, but there are a few sugars I ask you to memorize, not to be mean to you, but because you're going to need to know them for other classes. All right? So you can thank me later for that. All right? One is ribose. Ribose is a pentose, as you can see, meaning it has 5 carbons. It's probably the easiest one you're going to see the structure of. In the straight chain, it looks like that. All the hydroxyls are on the right. If we were to draw it in a ring structure, what we would see is that we would have a 5-membered ring, and the 2 at the bottom would have both down. Okay? Deoxyribose is related to ribose, and technically it's not a saccharide because it's missing an oxygen. The oxygen on number 2 is missing. Okay? But we mention it here because ribose is the sugar that we find in RNA. Deoxyribose is the sugar that we find in DNA. Okay? The only difference between them is that deoxyribose, deoxy means lacks oxygen, deoxyribose is missing an oxygen compared to ribose. All right, so you should know the structure of ribose. And this is both in the ring and in the straight chain form. This is true for all of these sugars. You need to know the structure of glucose in the ring and the straight chain. You need to know the structure of galactose in the ring and the straight chain. And you need to know the structure of fructose. You'll notice that fructose is the only one up there that is a ketose. There's that ketone right there. Okay. Now, fructose is a little harder to get the structure of. And I'll come back. Let me come back to that. Here we go. Actually, that's the dumbest figure. Well, since I brought it up, I'll talk about it here. So, fructose, your book uses a structure for fructose that I absolutely detest. I thought I'd put my own structure up there, but I guess I hadn't done that. I'll fix that. All right. If you look at the ring structure for fructose, fructose can exist in at least 4 different forms. All right? And no, I'm not going to expect you to know all the forms. The 2 on the bottom are very rare. We're just going to throw them out. All right? The 2 on the top are the most common, and it has the hydroxyl down or it has the hydroxyl up, corresponding to alpha or beta. Okay? Actually, this structure isn't. No, this structure is fine. It's the sucrose structure that's bad. The structures that you have on top here are fine. Okay, I confused this. All right. So these structures you're responsible for knowing, and you'll notice that this is why I want you to always number your carbons. When you number your carbons, you'll see carbon number 1 is right there. Carbon number 2 is there. Carbon number 2 is the asymmetric carbon. It's the anomeric carbon that we created by closing that ring. Remember, I said that the anomeric carbon will always be the ketone or the aldehyde. This was the ketone originally. All right? So the alpha refers to the hydroxyl position off of that anomeric carbon. All right? Then we have this guy, this guy, this guy, and that's what fructose looks like. So you can discard the 2 off the bottom in terms of being required to know the structures of those. Now, the beauty of fructose, of course, is you know one of them, you know the other one, because it's like alpha and beta. The only difference is that anomeric carbon in terms of its overall structure. Okay. Now, one other term I need to introduce, as if you didn't have enough terms already, and those 2 terms are pyrans versus furans. All right? A pyran is actually a molecule that looks like this. That's its name and that's its structure. And a pyran is something that has, as you can see, a 6-membered ring with one of the members an oxygen. That may sound very much like what you saw for the structure of glucose in the ring form. Okay? And in fact, it was the structure of glucose in the ring form. So we can refer to glucose as an aldose. We can refer to it as a hexose. And we can also refer to it as a pyranose. Pyranose. So if I put them all together, I would say, well, I have an aldo-hexopyranose. Or a hexo-aldopyranose. I don't care how you stack them up. Okay? It doesn't really matter. All right? Furan, on the other hand, is something that has a 5-membered ring, one of which is the oxygen, and that's what you saw with fructose. Fructose is a furanose, for the most part. Fructose is a furanose. Specifically, it's a keto-hexofuranose. Okay. Now, this figure I kind of like because, and maybe this is addressing your question up here earlier, this figure I kind of like because it shows going from the straight chain form of glucose transitionally into a ring and points out that that transition can result in either the formation of an alpha or the formation of the beta configuration. This flipping back and forth can happen in solution quite readily. So if I take, for example, alpha-D-glucopyranose, and I put it into water, okay, what will happen is some of it's going to flip all the way back to here, and then some of it's going to flip all the way back up to here, meaning that over time, I will not have all alpha D-glucopyranose. As long as the anomeric carbon is unaltered, meaning that as long as we have a hydroxyl right there, this flipping back and forth can occur. Now that turns out to be very important. And the reason it's important is because the straight chain form has a very reactive molecule on it. That very reactive molecule is the aldehyde. You remember from your organic chemistry that aldehydes are very reactive. And glucose, as a consequence, when it's in solution, can be very reactive because it can go back to this straight chain form. And when it's in the straight chain form, it can, in fact, readily be oxidized. It can readily be oxidized in solution. Fructose is a ketose. Ketoses, you may remember from your organic chemistry, are not so readily oxidized. Everybody hears about high fructose corn syrup, and everybody hears about how bad high fructose corn syrup is for you. And then you think, well, why in the world does anybody bother to make high fructose corn syrup? The answer is partly because fructose, as a ketose, is more chemically stable than glucose is as an aldose. Converting things into fructose gives some stability. Okay. Here's fructose doing the same thing, going from the straight chain form into the ring structure. And it's only showing you the alpha form here. This fructose can also, of course, form the beta as well. It's just not shown in this figure. And again, same thing can happen. This can go back to here, up to here, back to here, up to here. So if I started with all alpha D-fructose, okay, and put it in solution, over time, some of it would convert to the beta D-fructose form. Okay? Questions about that? Quiet group? Yeah.
Why is that syrup bad for you?
Why what? Why is high fructose corn syrup bad for you? That's a very good question. In fact, when I give you a lecture in just a little bit, I'm going to tell you why I think, this is Kevin Ahern's theory of high fructose corn syrup, about why high fructose corn syrup is bad for you. And I think I'll convince you, but let me save it until we get talking about metabolism, okay? Okay. It's related to obesity. Okay. Here's what all those sugars look like in rings. So there's ribose in the ring. I told you, very simple. See the OHs? Very simple to memorize. Glucose, fructose, galactose. You don't need to know mannose, but galactose you should know in the ring and the straight chain form. Okay. All right. Now, interestingly, and I trust you recall this from your, oh, that was a good job, from your organic chemistry, is that when we look at these sugars in a spatial view, all right, we discover that there's a little bit of twist and so forth that can happen with these bonds. And when these twists and so forth happens, there's actually two different conformations that a given form of a sugar can exist in. So even though, and these are not alpha, beta, this is both within the beta, for example. There's two different forms that a beta sugar could exist in. Okay? One form is known as the chair, and one form is known as the boat. All right? And it really relates to the positioning of the hydroxyls. You see, here's the hydroxyl on carbon number 6 over here. Here's the hydroxyl on carbon number 1 over here. This is called the chair because it sort of looks to some people like a chaise lounge. Here's the back. Here's where you put your feet down here. Okay? By contrast, the boat looks more like this. We have the carbon number 6. We have the carbon number 1 has been raised, as we see here. And there's actually interaction. There's actually steric hindrance that happens as a result of this. These guys can bump into each other. All right? Well, the boat form turns out it doesn't bump into each other so much that it doesn't form. It actually does form. But if we were to compare the two, the chair form would predominate because it would have less problem with interaction like this guy does. So boat and chair forms are actually different conformations of the same sugar. And it happens as a result of twisting of bonds that are occurring within that ring structure. Okay. That happens for hexoses. It also happens for pentoses. Now, pentoses are harder to understand, and we don't even give terms for the pentoses. So I'm just showing you these. You're not responsible for these, okay? But these guys look to me sort of like an envelope, all right? There's the lid of the or the flap of the envelope right there. There's the body of the envelope. And we can see that the envelope has been flattened out over here, okay, for what that's worth. But I just show you that for your own information. Okay. Now, let's come to some chemical reactivity. That's what I referred to earlier. And understanding the chemical reactivity of these guys helps us to understand, for example, why somebody wants to make high fructose corn syrup. All right? Here's the structure of alpha-D glucose. Okay? Glucose is the most abundant sugar on the face of the earth, period. Nothing's even close. Okay? Glucose is metabolized by virtually every organism on the face of the earth. And on top of that, as if that weren't enough, plants use it for making cell walls. And so glucose is everywhere. All right? Glucose can go from the ring structure, as I mentioned earlier, into the straight chain form. And the straight chain form has an aldehyde. This is not an aldehyde over here. Okay? This is a hemiacetal. This is an aldehyde. Aldehydes are reactive, meaning that they can readily be oxidized. Well, if they can be oxidized, we think about oxidation and reduction always happening in pairs. Glucose gets oxidized, something else must be getting reduced. And in fact, one of the ways we can measure this is if we put a solution of copper in the presence of glucose, that copper will get reduced. That is, it will gain electrons from glucose and move from being copper plus 2 to copper plus 1. This changes the color of the solution that contains that copper. The color will change. We can tell very readily if we have a sugar that can be readily oxidized by simply putting it in the presence of copper plus 2. We look for a color change. Voila, we have a sugar that can be readily oxidized. A sugar that can be readily oxidized is referred to as a reducing sugar. Okay? So glucose is a reducing sugar because it's reducing something else. In this case, it's reducing copper and causing a color change. Glucose turns out to be a very good reducing sugar. Fructose is not nearly so good. It's not nearly as reactive. It doesn't do as good of a job of reducing copper as glucose does. Okay? That's everybody looking at it. Is there a problem with the figure? Just looking at it. Okay. Now, what's happening when glucose is getting oxidized is it's going from an aldehyde to an acid. Again, organic chemistry. When you oxidize an aldehyde, you get an acid. Okay? And this is an acid form of glucose. Okay? This, I believe, if I recall correctly, is called gluconic acid. You're not responsible for that. There's others called glucuronic acid, different acid forms of glucose that we find in our body. Okay. Not all sugars are simple sugars. Some sugars are what we call complex sugars, meaning that they've had other things attached to them. And these more complex sugars we see up above. First of all, on the left, we see an L sugar. There's L-fucose, and look where that methyl group is. Notice it has a methyl group and not a CH2OH. Okay? So it's quite different from the things that we've seen. L-fucose we frequently find in membranes. Here's a sugar that is a fairly common sugar. It's called N-acetyl-galactosamine. And all it has is galactose with an amine and an acetyl group attached to it. That's the N N-acetyl-galactosamine. Okay, they don't have the N on there, but that's what it's called, N-acetyl-galactosamine. Here's N-acetyl-glucosamine, meaning we started with glucose and we added the same thing. Okay? We'll see later that N-acetyl-glucosamine is a monomeric unit of a polysaccharide known as chitin. Chitin is the insect exoskeleton. Okay. All right. Here's sialic acid. No, I'm not asking you to memorize the structure. These aren't anything you memorize the structure of. Right? But I'm just showing them to you because they are modified forms of the sugars that we see out there. Sialic acid is actually a fairly complex monosaccharide. You can see that it's got this guy attached to carbon number 5, and then it's got a separate R group that's actually fairly large over here. We won't worry too much about that, but just so you're aware of them. All right, now, when we take a sugar and we chemically modify it, okay, there's a variety of things that we can do to it, one of which is we can create what's called a glycoside. Okay? So I need to inform you about what that means, a glycoside. A glycoside is a sugar that has had an alteration to its anomeric hydroxyl group. Glycoside is a sugar that's had an alteration to its anomeric hydroxyl group. Okay? Well, what you see on the screen is something that we refer to as a disaccharide. I talked about monosaccharides before. I said they had one sugar. This guy has two sugars. Here is a glucose, and here is a glucose. And they've been joined together chemically through the anomeric hydroxyl of one of them. Okay? This modified anomeric hydroxyl creates this guy as a glycoside. And the bond is referred to as a glycosidic bond because that's what we find in a glycoside. Now, you should notice the naming here. The naming says this is alpha-1,4 glycosidic bond. Here's the glucose. This glucose is in the alpha configuration, meaning the hydroxyl was going down. That going down hydroxyl was linked to the down of a carbon number 4 hydroxyl. So we had alpha-1,4 glycosidic bond. That's what was there. Looking at this guy, I have a question for you. Is this guy going to be a reducing sugar or not be a reducing sugar? I hear some no's. Do I hear any yeses?
The last one is a no. The right one is a yes.
So his answer is yes and no. Okay, so I've got a no, I've got a yes and no. Do I have any yeses? The answer is yes. Okay? And the reason the answer is yes is it only takes one to make it be a reducing sugar. This guy can still go into the straight chain form. This guy can still become an aldehyde, and this guy can still become oxidized. Once I've altered the anomeric hydroxyl can't. So he's actually right. This one, no. This one, yes. But overall, the molecule is a yes because if any part of it can become oxidized, then it is a reducing sugar. Now, glycosides are made in a variety of ways. This is one that's made in making a disaccharide. We'll see other ways in which they can form. Okay? Here, actually, this wasn't what I wanted to show you. But if I were to, for example, put, let's say, a phosphate on this hydroxyl right here, I would create a glycoside. It would no longer be an anomeric, no longer be a reducing sugar. And it can no longer go back into the straight chain. Once I've altered that anomeric hydroxyl group, I prevent it from going back into the straight chain, which means this guy will no longer be a reducing sugar. Yes? It's always the hydroxyl of the anomeric carbon. Okay? I might put a methyl group on here. Same effect. If I do any alteration to that anomeric hydroxyl, then I've created a glycoside. Okay. This figure was simply to show you various alterations of sugars chemically formed. We'll talk a lot about this guy later. This is the first intermediate that's made in the breakdown of glucose 6-phosphate. Okay. How are we doing there? Okay. You guys up for a joke? A little dry today, huh? Okay. So, there's this lady, okay? And this lady goes to the pet store, and she goes to the pet store and she tells the pet store owner that it's her husband's birthday, right? And she wants to get something unusual for her husband, right? And the pet store owner says, okay, no problem. So he goes out and he brings out this enormous boa constrictor, right? And she looks at it and says, I don't know if I want to have that for my husband for his birthday. Okay, so he takes the boa constrictor away. He comes back and he's got this gigantic iguana on a leash. He trots it out here and says, how about this, you know? And she says, I don't think I want that around the house, you know? Oh, God, you know. So he goes and he brings out, 6 or 7 animals. And every time he brings out an animal, it's the same thing, you know. I just don't think so. And he finally says, all right, lady. He says, I think I know what you want. She says, what's that? And he says, you see that little bird sitting on that perch over there? And the lady looks across the room, and there's this little dinky bird sitting on the perch, just, doing its thing, right? And he says, that bird, he says, is a crunch bird. And she says, what's a crunch bird? He says, watch this. And so he says, crunch bird. He says, chair. And the crunch bird jumps up off of the perch, flies over to this chair, and demolishes the chair in front of her eyes. She's amazed. She says, I can't believe that. He says, you ain't seen nothing, lady. He says, crunch bird, desk. And there's an even bigger desk, and in a matter of a minute, it's sawdust right in front of her eyes. And she said, oh my God. She says, I've got to have this for my husband. He'll love it. So she buys the bird, she puts it on her finger, and she walks home. She walks in the door, and she says, honey, I got something for you. He's sitting there reading the paper, and he doesn't want to have anything to do with it. He goes, what is it? He goes, it's a crunch bird. He looks at it and says, crunch bird my ass. So much for the crunch bird. Okay, that'll wake everybody up now, right? All right. I'll tell you what, we'll do disaccharides, and then we will do a song. How about that? All right? Disaccharides. So, disaccharides, as I noted, are made by joining 2 sugars together. You already saw one disaccharide earlier. Now I'm going to name some disaccharides for you. Okay? The most common disaccharide that we talk about is sucrose. Sucrose is known as table sugar. Sucrose this is the one that I was thinking if I had the bad figure. The book has a terrible figure. I don't want you to use this figure. Don't. Underline, do not use this figure. I'll show you a better figure in a second. Okay? The reason it's bad is they've not drawn it in a natural configuration. They tried to save space, and they squeezed it onto this figure, and in consequence, students get confused by it. You'll see why in a second when I show you the actual one. But let me tell you about it. Sucrose is made by joining a glucose in the alpha configuration to a sucrose in the beta configuration.
Sorry, for what? Fructose.
What did I say? Fructose, I'm sorry. Sucrose is made by joining a glucose in an alpha to a fructose in the beta. Sorry. All right? Both of the anomeric carbons get tied up. That is, the anomeric carbon of glucose is joined to the anomeric carbon of fructose via an oxygen. It's what I like to think of as a diglycoside, meaning 2 glycosides. Okay? Now, I'm going to show you a different figure for that in a second. So as I said, don't copy this figure down. Lactose is another disaccharide. Lactose is also known as milk sugar. It's found in milk, okay? And it's made by joining a galactose, shown on the left, to a glucose, shown on the right. The linkage is a beta 1,4 linkage. There's the beta going up. Okay? And that's lactose. Maltose is very simple. It's actually what you saw earlier. It's two glucoses joined by an alpha 1,4 linkage. All right. So based on what I told you, I hope that you would recognize that since this is a diglycoside, sucrose is not a reducing sugar. There's no free anomeric carbon. Both of the anomeric carbons have been altered. On the other hand, lactose, which still has an anomeric hydroxyl free, is a reducing sugar. And maltose, which also has an anomeric hydroxyl free, is also a reducing sugar. Now, let me show you the better structure for sucrose. Notice I said stupid figure for sucrose. A better one is here. This high-quality art production was produced by me, and it actually shows you the way the thing looks. This is the structure I want you to know. All right? There's glucose on top. There's fructose on the bottom. You can see that there's the alpha part of the glucose. There's the beta part of the fructose. It's very simple. Publishers, for some reason, like to save ink and don't want to put that correct structure in books. I never understood why that's the case. Okay? I see everybody writing that structure down. If the figures are available before class, you can actually... Any questions about that? Okay, I've got a short song to summarize everything you've learned so far. And it's very easy to sing. I want to hear you today, right? Hark the sucrose. Carbohydrates all should sing. Glory to the Hayworth ring. Anomeric carbons hide when they're in a glycoside. Glucopyranose is there in the boat or in the chair. Alpha, beta, D, and L. Diastereomer hell. Alpha, beta, D, and L. Diastereomer hell. See you guys on Friday.